vertices. And once you have those three pieces of information, then you can start making these pretty pictures. But this is the minimal amount of information that's necessary to describe the surface mesh. And of course, once you have all of this information, then you can start computing distances, because you can just say, okay, how far is it from there to there, where I already know the x, y, z for this one, and then for that one, and for that one, and for that one, and then you can just add up the distances. You can also compute areas, because you know that what the area of the triangle is. Once you have the 3 points of the triangle and you have the x, y, z on the triangle, then you can compute the area of the triangle. And if you know that all of these triangles are part of superior temporal gyrus, then you can compute the area of superior temporal gyrus. This is also going to allow us to compute things like curvature and thickness, which I'll get into in a couple of slides. So just to reemphasize this contrast between working with volumes and working with surfaces. So if you're used to working with volumes, volumes are just a uniform grid. So you have some delta x, delta y, delta z, and that's how big your voxel is. And this is uniform, and the voxels are the same size regardless of whether you're looking in the front of the brain or the back of the brain, or the top or the bottom, or whatever. As opposed to a surface model where we have non-uniform sampling. So the triangles might be much smaller in some part of the brain than in another part of the brain, or the distances between these vertices. The voxel is an intersection of these uniform grid lines, and a vertex is an intersection of the triangles. A volume has some number of columns, some number of rows, some number of slices, whereas each vertex has an index. So if there are 160,000 vertices, then you can think of these vertices as just a list, essentially, of 160,000 numbers. Voxel has a certain size and a certain distance between them. And here we just have distances between the vertices, which are approximately, for FreeSurfer, approximately 1 millimeter. Every voxel can be assigned a value. For example, here I've assigned each voxel a value equal to its T1 weighted image. Each vertex can be assigned a value which could be something like its thickness value, or its curvature value, or say an fMRI value, or a PET value, or an EEG value, or an MEG value. Anything that you can think of being on the vertex, you can assign that vertex a value. But it doesn't necessarily have to have a value associated with it. And then finally, each voxel has an XYZ coordinate that tells you where it happens to be inside of this particular brain. And so we also have an XYZ coordinate. And this is how you map volumes and surfaces together, is that the surface should reflect the geometry of the brain, and that geometry is reflected by an XYZ coordinate in that particular brain. Yeah.
So if you do for the triangular volumes, do you normalize it to the triangle volume?
I'm not sure I understand.
I see. So if our triangles can be different sizes, and we assign it, say, a fMRI value, then do you normalize that fMRI value to the volume of your triangle?
Okay, yeah. So if you have a non-uniform grid, and so you have some triangles that are small and some triangles that are big, would you normalize like an fMRI value based upon the size of the triangle? And so for fMRI, you would not, because fMRI is just a point measure. If you were to look at, say, something like, so it's possible to map, say, area of one subject to area of another subject. So this would be like the area of a vertex or the volume of a vertex. So this would be something like doing a VBM study. And in VBM, you have this concept of modulating it or non-modulating it. And so this is basically the question that we're asking. Should you modulate the value at the vertex based upon the area of the vertex? And most of the time, no, because most of the time you're just looking at these point measurements. But if you're doing, say, an area-based study or a volume-based study, then you would. But that's a little bit obscure.
Oh, sorry.
Just a quick
question. Do the vertices align between subjects? So is vertex 22 in the same position as vertex 22 on another subject?
The question is, do the vertices align across subjects? Is vertex number 22 for subject 1 the same as vertex number 22 for subject 2? The answer is no, unfortunately. Okay, so, oh, sorry. Question?
Do the areas having a shorter curvature have smaller triangles?
Sorry, can you speak up?
Yes. Do the areas having a shorter curvature have smaller triangles compared to the areas which are more flat?
Do areas that have a sharper curvature have a smaller inter-vertex distance?
So the answer is no. So there are tessellation schemes that will take advantage of that, and cover like a flat bit with a huge triangle. But our interest is not in just modeling the geometric properties of the cortex, which we could do equally well with huge triangles and flat regions, but in doing things like sampling fMRI data uniformly. And so we distribute the vertices relatively uniformly across the brain regardless of curvature.
Okay. So to get back to the pipeline here, so we've used the WM.MGZ to create this jagged or ridge volume that has a resolution that is 1 millimeter. But we know that the cortical surface has a lot more resolution to it, or has a lot more smoothness to it than 1 millimeter. And so we create something called the white surface, which takes as its source the ridge surface, which you can see in green here, which is really jagged, and it essentially smooths it out. But it's not just applying a smoothing kernel to it. What it's doing is that it's adjusting the location of where those vertices are in order to follow the T1 intensity gradient. And so it's not just going and kind of sanding things down. It's actually using the MR intensities to drive that surface into registration with the underlying MR. And so we call this the LH.white or RH.white. And so that's just another surface. And then finally we create the peel surface, in which we take the white surface and we nudge it out normally from the surface, from the white surface, until we see a bright to dark transition. So the white surface is going to go from something that's bright, which is white matter, to dark, which is gray matter. And then the peel surface is also going to go from something brighter to something darker. And so we just nudge the white surface out until we find that next boundary. And that's what gives us the peel surface. And a couple of things to note. One is that along the medial wall, so we create the surface as if it were kind of this closed two-dimensional structure, kind of like a sphere. But in reality, each hemisphere is analyzed separately. But each hemisphere does not have a closed surface of gray matter around it, because you have subcortical structures in the medial wall, and you have corpus callosum that has to go through to communicate with the other hemisphere. So there are areas along this closed surface where there's not actually any gray matter, because there's not actually any cortex there. And so this is just something to be aware of. So there are not errors in the surface along through here where it's going through cortex or going through hippocampus or amygdala. And so we generally, when we're doing processing, we have a way to mask out those areas that don't have cortex associated with them. The next step is inflation. And so here we've gone through the volume-based stream, we've extracted the surfaces, and we have a white surface and we have a peel surface. And this is the white surface that's being inflated. So you can see that when it's in its constricted mode, mostly what you're seeing is green, because those are gyri, and then the red parts are the sulci. But of course, when you inflate it, what happens is that you can see both the gyri and the sulci. And this is what the inflated image looks like. And you can immediately see why nature has decided to fold up the cortex, because if cortex were not folded up, then your head would be something like this. You know, we look like the monster from Aliens, like walking around. So this is why things end up getting folded. And this also gives you an idea of what a surface model is. You know, this is just a closed surface which has the topology similar to a sphere, and it just gets folded up, and then you can unfold it. So to create this inflated surface, we just nudge the vertices out even more. There are no intensity constraints. Not really. There are some distortion constraints as well. But this is the inflated surface. So at this point, we've gone through the volume stream, and so that is the bias correction, skull stripping, automatic volume segmentation, and white matter segmentation. We've used that white matter segmentation to create an enriched surface. From that, we generate a white surface and a peel surface, and those are all the anatomical surfaces. And then from those, we generate this inflated surface. So if you remember, a surface has 3 components to it. It has a list of vertices. It has the nearest neighbors of each one of the vertices, and each vertex has an X, Y, and Z. And so that's what the LH ridge, the LH white, the LH peel are, as well as the RH versions of that. So each one of the surfaces has the same number of vertices, and they more or less correspond. So vertex 50 in the white more or less corresponds to vertex 50 in the peel. So that way, as you go from one surface to the other surface, there's some consistency there. So as Bruce described before, you can compute thickness values. So at every vertex, it's the distance from the vertex on the white to the vertex on the peel. And we go backwards and forwards to compute the average vertex. So this is a number that can be assigned to every vertex, and then we can render it again on this inflated surface. So you're going to see this inflated surface a lot. This is the thing that looks like a boxing glove. And if you're not used to seeing these things, this is going to be part of your education over the next 3 days, is to get used to being able to see what these surfaces look like, and to get some comfort in visualizing this boxing glove. For me, when I came into the field, I came into this, and so I don't really understand why people like looking at volume so much, but I understand that probably if you're not used to this, then it can become fairly disorienting to look at these types of surfaces. But hopefully you can get over that in the next couple of days. So this is just a surface rendering thickness. The color scale goes from 1.5 millimeters to I think 5 millimeters there. Can't see it. And so if you have bright yellow, it means that the thickness is somewhere around 5 millimeters. If it's dark red, it's 1.5 millimeters. And then if it's, you can see little patches of gray under there, meaning that it's less than 1.5.
These are native space measurements, they're not registered?
These are native space measurements. So everything that Recon all does is going to be in native space, and all volumes and areas and thicknesses are all native space. So if you were to cut this person's brain open and get a caliper out there and measure that, then that should be the same measurement that you would pick.
Which we've done.
Which we've done. Not to anybody. We can also compute curvature. So if you think, for each one of these different vertices, if you think of drawing a circle that would just come up and kiss the side of that of the surface, then the radius of that would be the radius of curvature. And so this is informative because it tells you basically what part of the fold you happen to be in. So if you're there at the crown, that's it requires a very, very small circle, so a very small radius of curvature. If you're further out, let's say in this big sulcus here, then that's going to be a big radius of curvature. And so we can assign a value to every vertex depending upon what its radius of curvature is, and we can render that as a colored image. In this case, green means gyrus and red means sulcus. and it's assigned, it has a sign plus or minus associated with it, depending upon whether it's a gyrus or it's a sulcus. And this is very useful because this codes the curvature pattern. So mostly people have a fairly, to a gross degree, people have a repeatable curvature pattern across subjects. And so we can use that to do this spherical registration. So this I'll talk more about tomorrow morning, but we basically take the inflated brain and we turn it into a sphere. And you can think of the sphere as being a planet, and you can think of gyri as being mountains on the planet, and sulci as being seas on the planet. And all these different planets look pretty similar. So everybody's left hemisphere looks pretty similar. Sorry, I don't know what that is. I'll just keep talking. Everybody's left hemisphere looks like a fairly similar planet with mountain formations and big oceans on it. So we can take those two and put them together, and rotate them back and forth, and try to align the mountains and try to align the valleys and the oceans, and get a better intersubject registration. So I'll talk about that tomorrow morning, I think. Then we do this automatic cortical parcellation, in which we take an atlas that's in our spherical space. So we create a common space that's somewhat like Telluride space, but only on the surface. And we have an atlas that's in that space, and then we can take that and we can map it back into the individual, and then we can fine-tune the boundaries so that it represents the labeling for that individual subject as best as possible. So these are the different types of overlays you can have. So now we've gone through essentially 3 different things here. The volume analysis, which is the bias correction, skull stripping, the automatic volume labeling, and the white matter extraction, or white matter segmentation. Then we've gone through the surface processing, where we take the white matter, dip it in chocolate, and we have a nice crust of a surface that's on the outside of that. That's called the ridge surface. We refine that to generate a white surface. We refine that to generate a peel surface, and then we inflate that. So that's the surface-based stream. And now that we have surfaces, we can start assigning values to those different labels. We can assign thickness values to it. We can assign curvature values to it. We can also assign labels to it. When we have fMRI, then we can assign fMRI labels to it. And these are called overlays. So the thickness, or sorry, a surface is 3 things: a list of vertices, their nearest neighbors, and the XYZ coordinates, and that's enough to define the mesh. An overlay is simply a value that I'm going to assign to each individual vertex, which then I can do statistics on, or then I can render as I've shown on this particular page. For the different surfaces, all of them have the same number of vertices associated with them. So regardless of whether you're looking at the white or the inflated or the peel, vertex 1000 is always vertex 1000. Finally, we create ROI summaries, and that's stored in this stats folder. And these are things that I'll go over more in the ROI talk tomorrow. But something like asec.stats would be everything inside of this box. So you would have something like left lateral ventricle for this particular subject had a volume of 7,347 cubic millimeters. And so all of the different regions that we have are going to be located in these different files. So we have a handful of files that we generate that are going to be inside of here. And so you can use these to go off and do ROI-based analysis. So if you want to look at, say, the difference in ventricular volume between normals and AD, for example.
Because the vertex 1000 is always vertex 1000, but for 2 different subjects, it doesn't always follow the same anatomical location.
Right. So again, vertex 1000 for subject 1 is not going to be the same as vertex 1000 in subject 2. We have to use this the surface-based work to get subjects into registration. And so I'll talk about that tomorrow morning as well.
So if I can just follow up on that, it's actually a design choice. So we have tools that could take the surfaces and make subject vertex 1000 be in the precentral gyrus or whatever in every subject. But the price that you would pay then would be that the vertex spacing is uniform in the atlas coordinates and not in subject coordinates. And so you would no longer be guaranteed to be uniformly sampling each subject. And you would introduce potential biases if people had larger superior temporal gyrus in one population than another. And so it's a pain from an engineering perspective to have the vertices be different across different subjects. But from a neuroscientific perspective, we think it's better, and that's why we do it.
And this last step is labeling. So you get an individual subject, do you transform it into the normalized space in order to, is that what you do?
No. So the question is on the labeling, exactly how do we do the labeling? Do we take the subject and transform it into the atlas space? We actually do the opposite. We have an atlas where the labels are located, and then we have the individual subject. But we have this registration between the two. So what we do is we take the labels from the atlas space, map them into the individual space, and then once they're in the individual space, then we fine tune them. Because the atlas labels for like the desiccant atlas are supposed to be gyro labels. They should go from the fundus of one sulcus to the fundus of another sulcus. And so if you just use the surface space mapping, you might not get that definition. It should be pretty close. But there's a second step which fine-tunes where that actual boundary is so that it's most accurate for that individual subject. So these RRI summaries, they're just statistics files, text files that you can go off and do kind of spreadsheet type of analyses on those things. Okay, so to summarize here, so you don't have the slide, I just made it last minute. You run recon all, which is something that you type at the command line. It goes off and runs for 6 hours to 18 hours or so. And when it's done, it creates this directory structure with a scripts directory, which has a recon log file in it, which is what you would send us if you have problems. It goes through a volume-based stream, and it creates a folder called MRI, and that's where the result of the bias correction, skull stripping, automatic volume labeling, and the white matter segmentation is stored. From the white matter segmentation, it steps off into the surface-based analysis. So if you remember, a surface is a list of vertices, their nearest neighbors, and the XYZ of the vertices, and that's what's needed to define the mesh. We have several different meshes that we use. We have the ridge, which is this jagged thing that follows 1 millimeter boundaries. We have a white surface, which should follow the boundary between white matter and cortical gray matter. We have a peel surface, which should follow the boundary for the outside. Then we have various overlays that it creates, including the thickness overlay. An overlay is just a value that is assigned to each vertex. Thickness overlay, a curvature overlay, and then a whole bunch of different overlays in there which you can explore. It registers the individual surface to an atlas from which it extracts labels and applies those labels to the individual subject and fine-tunes them. And the result of that is stored in this label folder. And then finally, it generates statistics based upon both the volume and the surface space stream. So the statistics are things like the volume of hippocampus, or the area of superior temporal gyrus, or the average thickness of caliper sulcus. To get FreeSurfer, so hopefully when you're done with this and you go back to wherever you came from, you'll be all enthusiastic about analyzing your data in FreeSurfer. So you can go to surfer.nmr.mgh.harvard.edu. You need to register, and the registration is important because when we go to funding agencies and say, look, FreeSurfer is really great, look at how many people are using it. So if you register it, then we get an extra little point with NIH, and hopefully they'll continue to fund us to do these kinds of things. So you register there, you download, and you can sign up for the mailing list. There's a FreeSurfer wiki, which is what you're going to be working from during the tutorials, but there's a ton of information, probably over 1,000 pages of documentation. We run under Linux, we run under Mac. We don't really run under Windows, but if you have a Linux virtual box for Windows, it will run under that. But it's not going to be a Windows environment. And it's going to be installed under FreeSurfer Home, which is another one of these environment variables. As Bruce went over earlier, there's several different ways to get answers to your questions. One is in our wiki, you can search for things. The other is you can go to the mail archive. So it's a very active mailing list, probably 10 or 20 different posts every day. And so probably somebody has had your question. And so you can go into the mail archive and you can type in a search term and probably you'll get a couple of hits, maybe even an answer. If you don't, you can send questions to surfer@nmr.mgh.harvard.edu. Then it will come to us. Please don't send email to us personally for two reasons. One is to have our own personal inbox flooded by all this FreeSurfer stuff is a little bit annoying. The other thing is that even though one of us might be up here appearing to be an expert on a particular subject, it might be that several other people can answer your particular question. And so it doesn't have to come to me, and it doesn't have to come to Bruce, or it doesn't have to come to Allison, that there are a host of other people who know about FreeSurfer, maybe even including one of you. So don't be afraid to answer questions that are posted to the list just because you're not one of the FreeSurfer developers. Finally, a way to get help is with individual commands. You can often type a -help after. So if you just type the command and hit return, it'll give you a list of different options, and if you give it -help, then it might give you even more information. So these are 4 ways that you can get help with FreeSurfer. And so I think I'm about out of time here. So I don't think I'll go over the tutorial tips right now, since you won't be doing tutorials until about 7 o'clock, I think. Okay, any other questions before you go to break? Question back?
In measuring the cortical thickness, the corresponding voxels between the one method and the file method.
Sorry, can you speak up?
Okay. In measuring the cortical thickness, do we measure the distance between the corresponding vertices between the hollow surface and the white surface? Do these corresponding vertices always the closest ones, or we have to measure we compute the closest spatial vertices?
So the question is, how do we compute the thickness? Do we just say, okay, here's Vertex 1000 and the white surface and the Vertex 1000 peel surface, and they each have an X, Y, Z, and so can we just compute that distance? That is not quite accurate, and the way that we do it is a little bit more complicated. So we'll start at the white surface, and we can compute the normal, and we march along that normal until we hit the peel surface, which it might not run through a vertex, it might just run through a triangle.
It's not actually the normal.
It's not the normal.
No, so normal doesn't work.
Do you want to?
Sure. Sorry, so actually using the thickness normal, the surface normal for computing thickness doesn't work. So if for example, you had the surface look like 2 flat U's, right? So the white matter surface was a big U, and the peel surface was a smaller U, then in kind of the outer edge of the flat U on the white matter surface, the surface normal will never intersect the peel surface, right? The thickness will just go on to infinity. So what we do instead is we do a search from each vertex on one surface. We search the region on the other surface for the closest point. And then we do that symmetrically. So we start on the white surface, we find the closest point on the peel surface for that vertex, and then we start on the peel surface, we find the closest point on the white surface, and we average those two.
How does that work in atrophy? You expect the cortical thickness to be smaller, right?
Yeah.
But you still from the tire to the white matter, you might have the same distance. Am I missing that?
No, I don't think that's the case. I think in cases of atrophy, we detect it robustly. So if the distance is less, then the distance is less.
Okay, but what if the wires have not changed? It's just the tissue inside it different? Because of
So that's a different question.
mush.
Right. So sure.
Can you repeat the question?
So the question is, I think, what if the signal properties of the tissue have changed,
Yes.
but not the geometric properties? And so we have a set of tools for characterizing that as well. Actually, Doug has done most of the work on that with David Salat. And that's an interesting biological question. You could look, so as Doug showed, you can essentially sample any cortical property onto the surface and do a surface-based analysis of it. And one of the things you can sample is the T1 weighted intensity. We've done a bunch on that, and perhaps somewhat more interestingly, we look at the local gray-white contrast, because we find that both white matter, like subjacent white matter, and cortical gray matter change intensities with things like aging or Alzheimer's disease. But that analysis is pretty straightforward, right?
So how do you calculate a cortical thickness of one RI? Do you take the average of all the?
How do we compute the cortical thickness of an ROI? So we just do a straight average of them. My experience, like we could do an area weighted average of cortical thickness, but my experience when I've looked at that is that it generates almost exactly the same, almost the same number.
So that means I can also, if I want to calculate the average cortical thickness of a bigger region of several RIs, I can also just take the average of those RIs.
In that case, I might do an area weighted average. So the question is if you had say 3 different ROIs and you wanted to compute the average thickness over those 3 ROIs, you could have one ROI that would be very, very small, like interrhinal cortex or something. It's tiny, and another one which would be really big, like superior terminal gyrus. And so you would probably want to, if you just wanted the straight average, then I would do an area weighted average across the ROS.
So which variable you will use for the average, the weight in that case?
The area of the ROI.
The area?
Yeah, so we produce average thickness for each ROI, and we produce area of the ROI in the stats tables. So both of those numbers are there, so you could put those into your program.
What is the difference between the sulk and the curve surface of the legs?
What's the difference between the curve and the sulk? Is that where your question is? Okay. So this one is the sulk, and this one is the curve. And so the curve is this curvature measurement that I described earlier that's just looking at basically how sharply is the fold turning in 3-dimensional space. The sulk is computed differently. It is basically if you ignore the breathing brain here and look at this one. So the sulk is the distance that a vertex has to travel from the white surface to where it gets inflated to. That's right, more or less.
Yeah, so it's the sign distance during inflation. So if you're of the dot product between the movement vector and the surface normal. So if you're deep in sulcal, as you move outwards, you're moving along the surface normal, the dot product is positive. You sum that up, you get a big positive number. If you're gyral, then you're generally moving inwards. The dot product with the surface normal is negative. And as you sum that up over inflation, it'll be negative. And so the nice thing about sulk, as you can see on one slide, that one, is that it reveals the kind of large-scale geometric features of the cortex better than the mean curvature, which is shown in the middle there. Right, so the mean curvature has all this high-frequency information that is probably too much for basing things like cross-subject registration, whereas the sulk highlights these deep phylogenetically and evolutionary old structures, that we know exist in every subject, the Sylvian fissure, the calcarein, the central sulcus, these kinds of things. And so it's a better basis for doing intersubject registration than the curve. I mean, we actually use curve information, but the primary driving force is the sulk.
Its vertex is either sulcus or gyrus, so it's 1s or 0s or 1s and 2s?
No, it's a continuous measure.
Okay.
Calculating the curvature, there was a circle that was fitting. Along the surface there can be multiple circles along different directions. Is it actually really a sphere that is fitted, or it's the smallest circle along the one direction?
So for any two Oh, so the question was, are we really fitting a circle when we compute the curvature of the surface? And the answer is no. The answer is that we're formally, in a mathematical sense, we're computing the mean curvature of the surface. And one interpretation of the mean curvature is the smallest inscribed sphere. And for any two-dimensional surface, there are actually two principal curvatures you can compute. The curvature in the direction of maximum curvature and the curvature in the dimension of minimal curvature. And those are kind of two differential geometric measures that we automatically compute that are related to the kind of local differential properties of the surface. But you can think of them as inscribed circles. So I don't know if, was that clear?
Their average,
I'm sorry?
and then their average,
The mean curvature is the average. We could compute other properties, like the Gaussian curvature is the product of the two principal curvatures. But the mean curvature is kind of more geometrically informative, and it's what you see up here in the middle on the top.
I was wondering, the DICOM data might contain personal information or patient's name. Is there any tool for clearing that kind of information?
So the question is, the comment is that there's could be personally identifiable information inside of a DICOM file. And I was wondering if there's a way to anonymize. So when we convert to the MGC format, all that information is stripped away. So there's no personal, there's no header information that is personal or identifiable once it's converted to MGC or Nifty for that matter.
Could it be in every column slot?
It might be. Like if you were to go and enter the subject's name, like the scanner, sit down at the console and actually enter the subject's name, then that might be printed out and then be captured in the log file. Okay, so I think our
We have a break.
we have a break until right now. So why don't we take a 15-minute break?